Phylogenetic reconstruction is a really core part of evolutionary biology, but building phylogenies is actually very difficult. Now, when I say it's difficult, I don't mean that they're hard to do in the present day. It's actually very easy to build a phylogeny. If you have sequence data, you can plug it into computer programs and you'll get a phylogeny out the other end. That's very easy to do. However, what's going on inside the programs is actually very difficult. And it's very hard to build a phylogeny for a group of organisms that is the same using lots of different genes or using lots of different characters. Now, why is it difficult to build phylogenies? Why is it hard to make sense of natural patterns of variation among different species in the natural world using phylogenies? Well, phylogeny building would be easy if we saw a pattern of variation like this. And I'm going to use letters here to depict different characters. So a letter A, for example, might be a certain type of leaf, or it might be a certain type of wing, or it might be a certain coloration pattern of a petal, or something like that, some kind of character that an organism has, whether it's a plant or an animal or a microbe. And if we found that if we were looking at 5 different organisms, if 2 of them had character A, 2 of them and 3 of them had character B, if we were just looking at that character, we'd think these 2 organisms, species 1 and 2, are more closely related to each other than they are to these 3 species, and these 3 species are all closely related to each other. So we might depict that like this. We could tell something about their relationships. And then we might find, if we look at another character, well, here's character C, and that is just found here and here. It's only found in things that already have B. And that's really nice, because now we can tell these 2 are more closely related than this one, and so we can change our tree and we can make it like this. Here we have a nice nested hierarchy, and maybe we look at another character, character D, and character D is only found in things that already have A, and then perhaps character E is also only found in things that have A, but it's only found in one of them. We might then find another character, let's call this F, and it turns out F is found in all of these. So F is suggesting that all of these are related. And so we can build a phylogeny like that. And what we have here in our characters is a very nice nested hierarchy. It's a hierarchy where everything that has A also has D, and there are no characters that sort of cross over between groups. So for example, if this wasn't a nested hierarchy, what we might find is that character G is found here and also here. Now this poses a problem for us, because G is suggesting that this organism and this organism are more closely related. However, characters A and D and not F, because F is found everywhere. Characters A and D and B and C are suggesting that this organism and this organism are not closely related. So character G isn't fitting into a neat nested hierarchy. Now in this case we've only got one character that's not fitting in the hierarchy, and so it seems to us that the evolutionary tree that we've drawn is probably correct, and this character has evolved convergently. We sometimes call that homoplasy. So we might say that G is a homoplasy. Whereas the characters that fit the tree very nicely, we call them homologies. Homologies are easy to interpret in an evolutionary framework, because homologies are similarities that are due to shared common ancestry. Homoplasies are similarities that are not due to common ancestry. Now, if we only have a little bit of homoplasy, then it's easy to build a phylogeny based on the nested hierarchy of the pattern of variation that we see. However, homoplasy is very, very common. When we look at real organisms in real life and compare their similarities and differences, we find a real hodgepodge of different characters. We don't find characters that neatly fit into a nested hierarchy. We find this jumbling up of different characters all throughout life. And this makes phylogenies very difficult to build. This means that it's very hard to work out what is a homology and what is a homoplasy. And so for any set of organisms, there are very often more than one tree that can fit the pattern of variation. It just depends what you think of as a homology and what you think of as a homoplasy. Now, that was very evident to people when they were looking at morphology. Initially, as we saw in the first lecture, phylogenies were built based on morphological variation. And there people very quickly discovered the pattern of variation doesn't fit neatly into a nested hierarchy. You have lots of characters that are very hard to fit into a phylogeny. And so people thought, well, maybe when we start looking at the genetic material, maybe when we start looking at DNA, then we'll find there's much less homoplasy. The thinking was, well, morphological characters could have evolved convergently due to the same selective pressure. But surely that's much less likely to happen with DNA data, especially if we look at parts of the genome that are evolving in a neutral fashion. And then when people started to sequence genes and build phylogenies, when they were just using one gene, they often found they could build a fairly nice phylogeny. It was fairly easy to fit the data to it. However, when we started sequencing more and more genes, we soon began to discover that different genes give different phylogenetic trees. So this problem of homoplasy, this problem that patterns of variation don't fit neatly into a nested hierarchy, came back. Now, of course, the more and the more genes you sequence, eventually you hope that the weight of evidence will go one way or the other and allow you to build a phylogenetic tree that's fully resolved. But often that doesn't happen, and often we are trying to weigh up statistically the different probabilities of different trees. And so you get different trees according to what data set you use, which genes you use, which models you use of evolution, what statistical methods you use to analyse the trees. You get lots of different results. And all of this is because the characters in life are all jumbled up in such a way that they don't fit neatly into a nested hierarchy. And this is what makes phylogenetics a really difficult area.